Good morning. As we all know, with the ever-growing population, the food source available to us has gone through many grandiose modifications starting in about the 70s. They started using different types of fertilizers and pesticides, which granted did produce an increase in yield, but then at a great cost to our environment. Upon noticing this, we then shifted to something that was a little bit more INC in the nature of modifying the genetic makeup, which would produce a yield that had a much higher caloric value. Taking a sidestep, we'll look at Dr. Thane Papke, who studied here at Augsburg during his undergrad years, went out and continued research into his great interest of what is now considered to be the domain of archaea, which is a single-celled organism which carries its DNA without, or I should say, not contained within a nuclear envelope. This fascinated him for many reasons, primarily that the diversity in something that was self-replicated, meaning that it didn't follow the conventional means of having a male and a female unite in order to have two different separate genes come together in order to create a different, or I should say, the enhanced version of the most suitable genes in their offspring. Studying this, he noticed using GFP, which are radio telemarkers, in which you can see fluorescent lights that indicate exactly where the chromosomes are within the organisms that he's studying. Early on, he noticed that the specific Heloarchaea that he was looking at carried 30 copies of its own gene. Now, looking at this, he was more interested in seeing how they would transfer their genes from one individual to another using methods we now understand not only are bits and pieces that are transferred from one particular individual cell to another, but then are also then self-spliced into themselves, which then can be transferred completely upon its quote-unquote offspring. But one thing he didn't expect and noticed that this specific archaea carried 30 copies of its genes. In conducting the experiment, he noticed that once the environmental sources of nutrition were depleted to a point where it had to start using its own resources in order to maintain or function survivability, he realized that this number of 30 copies slowly depleted down to 2, which is the bare minimum required for them to self-replicate. Now, upon looking into this, the GFP that showed up as really bright lights under specialized microscopes then indicated that not only did it carry these 30 copies, but that it would, throughout its lifespan, just secrete extracellular DNA, which then would be reabsorbed by either itself or those around it. But then something odd happened where it would start then using this and metabolizing this for sources of phosphorus, carbon, and nitrogen. He did a set of experiments starting with herring DNA, ending up with a modified set of Ebola DNA, which what indicated to him his next step in research was that there were certain identifiers along these units, along the DNA strand that were quote-unquote methylated, which were markers so that it could identify its own DNA opposed to other DNA that it wouldn't touch because it didn't contain these identified markers, which it would then tell the cell to either metabolize or maintain. Now, getting back to using these particular, or using this as a nutritional source for phosphorus, nitrogen, and carbon, you look at closer are the exact same elements that you'll find in what we use as human beings, not only as our quickest, most immediate source of available nutrition for energy conversion being glucose, and even in the way that we store it as glycogen. Looking at this shows that there is a great potential in utilizing this even as ourselves, not only as a source of our genetic material, the makeup that makes our cells, but is readily abundant in every living organism on the planet, and now shows promising signs that it could be a very highly potential source of nutrition for ourselves to supplement our own deficit given our own growing population.